Hey everybody, a little bit of a change of scenery today. I'm back in my office, back on campus, and ready to talk about what I think is a really exciting phase of understanding where the human genome map and our linkage analysis comes from. You'll notice I have my official coffee cup from Jergolitz Duck Farm, even though it has a rooster on it, because that was from the year of the rooster, the Chinese New Year back then. So we're really in a position to really start integrating and talking about advances in molecular biology and the biochemistry of the gene and how they all integrate to create the genetic map. And the first effort to create the genetic map was really in the early 1980s, and if you recall, this was actually before the Human Genome Project got started. Genome Project got started actually approximately a year or two later, and was not completed until 2001, but we'll talk a little bit more about that later on. Notably, Sir Alec Jeffreys is credited with the idea of looking at restriction fragment length polymorphisms. Now remember, polymorphisms in our traditional genetic sense is just when you have different forms of a gene, polymorphic, many forms, all right? And it was Alec, along with other researchers at the time, Alec recognized that these polymorphisms did not only occur in phenotypically observable traits, but there were polymorphisms for specific DNA sequences, and that you could see that those sequences are inherited just like any other Mendelian character, and figured out a way to annotate the RFLPs. So he really took advantage of something, a rather unique discovery in the 70s, which was an enzyme called a restriction endonuclease, and we'll talk about that in a moment. And he saw he could use these restriction endonucleases to distinguish among and between different DNA sequences. So he could look at these templates, digest them by the restriction endonucleases. Remember, endonuclease, okay, inside the nucleus or inside DNA. And he was able to create a power of discrimination using 6 different either enzymes and or DNA probes of about 1 in 10 to the 6th to 1 in the 10 to the 8th. So about 1 in a million DNA fingerprints. Let's back that up a moment. Meaning he could identify a DNA fingerprint that was unique to 1 in a million people. Okay? Now, of course, we have to talk a little bit about what a restriction endonuclease is. Endonuclease, within DNA. It is an enzyme that cuts strands of DNA at a restricted sequence of bases. So you can notice in our first BAMH1 endonuclease here, you could see it cuts between the G and the C, and the C and the G on the 3' to 5' and 5' to 3' strands, okay? And it creates kind of a jagged cut and cuts the DNA into 2 pieces. One of the things that you should notice is that there are over 350 different endonucleases, that is, they cut at 350 different DNA sequences, and they are all named after the bacterial species that they have been identified from. Now, we'll talk a little bit later on today specifically about Echo R1 and also HIN3. These are the two restriction enzymes that we'll be using in lab today for the when looking at how those enzymes digest plasmids from our E. coli transformations. So here's the idea about RFLPs. So here we see that we have a wild type DNA segment which is indicated at gene 1, and we'll just call it allele 1. This individual sequence has 4 different alleles indicated by 1, 2, 3, and 4, and the wild type has, and this is only using one single, sorry, phone's ringing again. This indicates that there is one restriction enzyme that cuts at the TCGA site. So we're just going to show a single strand of DNA, TCGA. Okay? And notice that when we look at this site, we've got TCGA. Notice that's TAGA, TCGA, TCGA. So this enzyme will cut at the C, between the C and the G. So there's a cut here, and there's a cut here. There would be a cut here, but this creates a very, very small piece of DNA. So here we have this, and notice here's our legend. We have a 4 kilobase bit of DNA, and it can be visualized, because remember, especially in the 80s, we had no ability to find these very small bits of DNA. They would attach a probe that specifically identified a sequence of DNA that existed between the 2 and 3 kilobyte stretch. So that means that if we took a homozygous wild type individual, okay, and we were to cut up the DNA with the restriction enzymes, it would result in a 4 kilobase bit of enzyme, all right, a 4 kilobase bit of enzyme, and we would see, if we were to put that and separate the DNA, put it on a gel, separate it, we would find a single 4 kilobase fragment. Now, why only a single band? Remember, everybody's diploid. It's because the individual's homozygous. So you only see one band, so we know it's a homozygous wild type. But now let's look at alleles 2, 3, and 4. Notably, in allele 2, okay, allele 2, notice that there is a mutation right here. So the mutation changes that TCGA to TTGA, which means there's no longer a cut here. The cut occurs a little bit downstream, so we have now a much larger piece of DNA. It's now 5 kilobases long. What if there's another kind of mutation, and this is allele 3? So we have a change here where TAGA goes to TCGA. Well, what happens? We end up with another site where the restriction enzyme can cut, and we end up with a much smaller bit of DNA, being somewhere in the neighborhood of about 2 to 2.5 kilobases. What if in the 4th allele we have a 0.5 kilobase or 500 base pair DNA insertion? This means that the wild type gene still cuts in exactly the same position here and here, and over here, but is much bigger because of the insertion of a fairly large stretch of DNA. All right? So now we've got 4 alleles, and if we extracted the DNA from 5 different individuals, you can see that 1-1 is a homozygous individual that has allele 1 and 1, which results in 1 band. An individual which is heterozygous for 1 and 2 has 2 bands, one being, of course, 4 kilobases, the other 5. And if we had a 2-3, we've got a 5 and a 2.5. Allele 3 and allele 4, we've got that 4.5 and 2.5. And of course, an individual that is homozygous for both alleles containing the 500 base pair insert is homozygous, and again, you only see the single band here. So this became a really exciting method to distinguish among genotypes based not on a particular phenotype, but rather based on a particular DNA sequence. So pretty cool stuff. Alec was involved in something called the Colin Pitchfork case. So imagine being able to take DNA from a human being, expose them to endonucleases, utilize the probes, and to utilize gel electrophoresis to separate out a DNA fingerprint that is a unique set of bands in a gel electrophoresis lane, a unique set of bands that could identify any individual. And it was in the early 1980s, specifically in 1983, a young woman left her home to visit a friend's house. She did not return. She was attacked and killed. Very scary at the time. It's always scary. But at the time, forensic analysis basically just was able to identify that the person who killed Linda had type A blood, and they were able to look at acid phosphatase levels in the semen remaining on the body that only matched 10% of the males. There were no other leads, there was no other evidence, and the case was left open, so there was no resolution. In '86, almost 3 years later, another young woman took a shortcut instead of getting on her normal path home. Several days later was found, again, beaten, raped, and strangled. The modus operandi, or the MO, matched that of the first attack on Linda Mann and the first murder, and analysis of the semen samples identified, again, type A blood and a phosphatase level that matched 10% of males. At the time, the suspect was Dawn's boyfriend, whose name was Richard Buckland, seemed to know that Dawn was taking a shortcut home and admitted under questioning that he killed her but denied the first murder. A horrible, horrible situation. At that point in time, the idea of these RFL DNA fingerprints was published by Alec Jeffries. And Jeffries was able to obtain samples of the semen from both murders, subject them to RFLP analysis to see their DNA fingerprint, and compare them to the DNA fingerprint of Buckland. It turned out that both girls were killed by the same person, meaning those DNA fingerprints matched perfectly. However, those samples did not match a sample taken from Buckland. Really quite remarkable. This is the first case, not just of the use of DNA fingerprinting in a legal proceeding, but also the first person to have their innocence proven without a doubt by DNA fingerprinting. You may wonder, why in the world did he admit to the killings? And the answer really had to do with the difference in the legal systems between the U.S. system and the British system. While he was being interviewed, he was kept awake for 3 days without water, without food, and was constantly under the pressure of being questioned, and finally at the end just decided to admit it. Quite a stressful period of time. But now the problem came about. They knew that the same person committed these murders, but how to find them? Again, a little bit different than the U.S. system. It's a small village. 5,000 men in the village were asked to volunteer blood or saliva samples. It took Alec's lab 6 months, and they did not find any matches. So none of the men who lived in the town that they collected blood and saliva from matched. So it was really disappointing. However, a bit later on, Ian Kelly was at a pub, and he paid off his tab at the pub and also admitted to the bartender, after having several drinks, that he was given £200 to pretend he was the local baker, Colin Pitchfork, and gave a blood sample instead of the baker. You guessed it, in 1987, the police arrested Colin Pitchfork, took a blood sample, and it matched the 2 murdered women. So not only did we see that this particular case provided the first person proven of innocence, but also the first person that was caught and proven guilty on the basis of DNA fingerprinting. So really a very, very important case in the world of genetics and in the world of forensics. Now, you do have to think about this as being real limits. Part of the problem with RFLP is it requires a very large sample of high molecular weight DNA, around 50 to 100 nanograms, which is thousands of cells worth of DNA. And this is not at all ideal for forensic analysis, where you only find small and very often degraded samples, meaning you don't find entire strands of DNA. So what could possibly be done to improve this kind of an analysis? And we see here polymerase chain reaction to the rescue. Identified by Dr. Kary Mullis. Kary won the Nobel Prize for being the inventor of polymerase chain reaction, and basically utilizes different temperature phases carried out in a device called a thermocycler to amplify specific sections of DNA. There's a nice little graphic here showing you a conceptual idea of how DNA is separated. Remember from talking about the biochemistry of the double-stranded DNA, how can we separate? How do we break the hydrogen bonds that hold the 2 strands together? And the answer is we apply heat. The best way to break hydrogen bonds, apply heat. So we apply heat, the DNA strands separate, and then it's called polymerase chain reaction. So this utilizes a form of DNA polymerase. DNA polymerase. Remember that? In replication, what does DNA polymerase require? Think a minute. DNA polymerase, in order to undergo replication in a cell, has to have a primer, an RNA primer, which provides a landing strip for the DNA polymerase to land, and of course begins to work in both directions on the leading and lagging strands to create de novo strands of DNA. Well, in polymerase chain reaction, a DNA primer is used, and this primer exists in complement to an existing or known bit of DNA or a bit of sequence on the original DNA strands. Once it binds, as you lower the temperature, hydrogen bonding can begin again. We find these primers bind specifically to that sequence of DNA. DNA polymerase then is added, and at a slightly elevated temperature, DNA polymerase becomes activated, and it creates two new strands of DNA. And of course, as you continue to remake the temperature cycle, it goes over and over. Generally, these cycles can go between 25 and 35 times, depending upon the DNA sequence, depending on the enzyme, depending on a lot of different biochemical issues. Now, if you think about that, you could theoretically take a single strand of DNA and get 2 to the 30 copies of DNA by using polymerase chain reaction. Now notice one thing, I want you to take a peek. What made Mullis's discovery so impressive? And notice here, I don't say DNA polymerase. He used something called Taq polymerase. Taq polymerase. Didn't we just talk about something that was named with 3 letters when we talked about endonucleases and restriction endonucleases? Of course. So what do you think those 3 letters stand for? And if you guessed, the organism from which the polymerase was derived, you'd be absolutely right. And it turns out that Mullis studied archaebacteria. And in particular, he was interested in studying extremophiles. And the reason that this was important is several individuals had come up with the idea of doing PCR. Okay, several people had came up with the idea of doing PCR in a test tube. But unfortunately, during the process of raising the temperature of the DNA to 95 degrees, what happens to protein if you raise it to 95 degrees centigrade? Well, you've all boiled an egg. The proteins denature and, in fact, inactivate enzymes. So what was it that Mullis did that was so unique? And that is right here, the Taq. Okay? The Taq is for Thermophilus aquaticus. It is an extremophile, thermophile. It exists in hot water geysers. And if a hot water geyser does not get hot enough to inactivate the enzymes within that particular bacteria, well then Mullis said it will not inactivate the polymerase. So he extracted the gene for polymerase from the Thermophilus organism. He inserted it into E. coli. He made a transgenic E. coli, just as we've been talking about in laboratory. Cranked out milligrams worth of Taq polymerase, and eventually developed the conditions under which PCR could work. So really exciting development, and of course one that eventually won him the Nobel Prize. There's a link available in the notes which will allow you to go and see some of the controversy surrounding the Nobel Prize. It had a lot to do with the patenting of the process, the patenting of the gene for Taq polymerase, and a lot of the different companies that were involved in trying to utilize the PCR technique for a variety of different both forensic but also diagnostic assays. So one of the first forensic applications utilized a unique marker called the HLA-DQ alpha. This is part of the major histocompatibility complex. It is one of two proteins that is present in the human immune system. It is a marker of self that exists on the surface of all cells and exists in very, very large amount of variation. It has a very highly variable DNA sequence, which makes it particularly amenable to analysis by doing PCR to identify all the different variants. So instead of slicing and dicing DNA by using the restriction enzyme, we actually can use PCR to replicate each of the variants and identify them. So we have here, the other advantage is, of course, PCR theoretically only requires one molecule of DNA. So its sensitivity is much better in that it requires more than an order of magnitude less DNA. It also does not have to be high molecular weight DNA because it just relies on this very small stretch. And the power of discrimination is about still 1 in a million, which really is not good enough for databasing for forensic analysis. So one more little discovery. As time went on throughout the 80s, more and more was being identified about the human genome. And part of what was identified was only 2%, 2 to 3% of the human genetic code is actually coding for proteins. Actually coding for proteins. What does the rest of it do? Well, we still don't have an entire answer for that. But one unique feature of the non-coding bits of DNA is that they existed in what are called short tandem repeats. These short tandem repeats are sequences of 2 to 5 base pairs that repeat themselves multiple times throughout the chromosome. The one I've got here to show you is right here, is GATA, and in this case there are 4 GATA sequences in a row. This is a tetramer, and it turns out that there are many polymorphisms of STRs due to the number of copies of the repeated element. So some of us would have perhaps 4 copies of GATA all in a row. Others of us might have 16 or even 20 copies of GAT in a row. And it turns out that these are inherited just exactly the same as our restriction fragment polymorphisms, just exactly the same as any Mendelian phenotypic gene. So we can use now these short tandem repeats to identify unique polymorphisms based on DNA sequence, and because they are identified by PCR, you need very small amounts of DNA. It does not have to be high molecular weight DNA. It could be degraded, so good for blood spatter around a murder scene. And in fact, the current method of choice is to utilize autosomal short tandem repeats. These, again, require around the nanogram of DNA. We type 13 to 15 short tandem repeat loci, and the power of discrimination among these means we can find unique possibilities of approximately 10 to the 23rd power, meaning 1 to the 1, the ratio 1 to 10 to the 23rd power. That's a huge number. That's the number of atoms in a mole, just about, right? So the world population is only 10 to the 9th. So this means we're able to actually create a database where each individual is absolutely unique from one another. Absolutely unique. Here's what one of the sequences look like. Here, in fact, is the one we've been talking about. This is a short tandem repeat on chromosome 7. It is one of 13 that are currently used. Take a peek at the DNA sequence, classic way to look at DNA sequences, and how many repeats do we see in the sequence? Approximately 6 to 15 are found among humans. So there are, right, 6 through 15, there are 10 different alleles. So instead of using a simple mutation or even ABO blood groups, where we would have 3 different alleles, now we can look at an individual short tandem repeat that has 10 alleles, and if we use 13 of those short tandem repeats, well, we can create a very unique looking DNA fingerprint. And here is a great illustration, a graphic analysis of, here is a piece of DNA. This is John Doe's DNA. And here, of course, is Jim Doe's DNA. And you can see they now added the concept of V. So this is variable number of terminal repeats. These are short terminal repeats that vary in number of copies. Again, you can see that our variable number of terminal repeats here, we have 2 different loci for John at the locus 1 rather, 2 different sizes for locus 2, 2 different terminal repeats for locus 3. If we were to look at Jim Doe's DNA, he has 2 different sizes of DNA here, 2 different sizes, 2 different sizes, and you can count up the number of repeats. And what's really very, very nice about this is we begin to say, for example, if you look at the green individual here, we have just in John Doe, so we take a peek, there are 4 green boxes here, and of course in this one we've got 8 green boxes, so there are 8 terminal repeats. Jim Doe has 8 in the first section, 7 in the next, and you can see when we run it on a gel, all right, we see here is 8 in the green, here's 4 in the green, here's 8, and of course 7 for Jim Doe. And we can see very clearly why this begins to look like a fingerprint. We have 2 entirely different patterns of banding between the 2 individuals. Kind of cool. Of course, this is done by a machine in capillary electrophoresis. Let you look at this on your own. We don't need to spend too much time with this. We no longer do this by gel analysis. We actually do it by a machine that looks very much like a gas chromatograph, only it does polyacrylamide electrophoresis throughout a capillary tube. And it records the data by the amount of signal that is seen as a laser is shown through the capillary tube. And in our case, you still see, here's the 4 and the 8, and of course, we see this is 2 completely different individuals, 5 and 13 sticking with the gene. So we see completely different sets of nucleotide pairs. Of course, part of the problem is when we look at these are all autosomal. They're all autosomal. So we have to have some way of identifying sex, and the only way to do this is to have something on either the X chromosome or the Y chromosome that can be identified, and identified by PCR. So it turns out that there is a gene called Amelogenin, and again, yes, we got a polymerase chain reaction. It turns out Amelogenin is a single copy gene, and there is a homologue to Amelogenin on the X chromosome and on the Y chromosome. And strangely, intron 1, because remember, we are eukaryotes, intron 1 on the X chromosome is 106 base pairs, whereas intron 1 on the Y chromosome is 112 base pairs. This is because the X chromosome copy of Amelogenin has a 6-base pair deletion, and so we can run them on the gel and separate them, or we can run them in conjunction with our short tandem repeats or variable number of tandem repeat analyses. I should point out that all of these analyses can be done at the same time in a single reaction by mixing up all of the primers in one solution. So basically it happens very, very quickly. If we take a peek at what the data actually look like, you can see here we see the result of Amelogenin analysis in the first column, and these are actual data from the FBI for a victim's DNA, victim of an assault, the suspect's DNA, so you could see the male suspect assaulted a female, and there was DNA taken from the suspect and also from the victim. These are two different DNA samples from the same piece of evidence. So think about it, if this were, for example, somebody hit someone with a bat or a spatula or a hammer or whatever, one end would be proposed to be the victim, one end would have the suspect's DNA, and you could see very quickly they can differentiate between male and female. All subsequent columns, excuse me, all subsequent columns are from a different short tandem repeat, and you can see that the data proposed to come from the victim matches the DNA taken from the evidence, and also the suspect's DNA matches in every case, matches the DNA on the evidence. So we are really able to identify these individuals, and our odds of it being exhibited by someone else, that is, making a wrong decision of identification, are about 1 in 100 quadrillion. That's a pretty big number. We're not going to see that kind of a mistake very often. So this gives you an idea. This gives you an idea of the biochemical techniques and how they're applied to forensic evidence. In the next mini lecture, we're going to talk a little bit about modern applications, and there's even one further bit of molecular biology and biotechnology to talk about, and it applies very, very specifically to personal genomics and our current method of sequencing DNA. You guys take care, be safe, don't forget to mask up. I can try. We'll see you on the next one.